Good afternoon, and I'd like to welcome you to the Human Research Program panel this afternoon. As a reminder, please submit your questions using the tool if you have them during the panel. We'll hold all questions until the end, so be sure to note which specific speaker you're asking the question to. First up in the panel, I'd like to introduce Dr. Bill Pawlowski. Dr. Pawlowski is the director of NASA's Human Research Program. The program focuses on research and technology development aimed at mitigating risk to human health and performance during spaceflight. This program funds over 250 investigator teams working across multiple platforms, from the International Space Station to flight analogs, as well as government and academic labs. Dr. Pawlowski was a professor of human health and performance at the University of Houston prior to his current position, and prior to that was a longtime researcher in neuroscience at the Johnson Space Center. Dr. Pawlowski holds a PhD from Rensselaer Polytech Institute. Dr. Pawlowski will be speaking about the NASA Human Research Program mission and overview, as well as giving a brief introduction of the other speakers in the session. Now I'd like to hand over to Dr. Pawlowski.
Okay, thank you, Jennifer, and thank you for the organizers, for inviting us to come and speak at this meeting today. Most of us from HRP come occasionally to this meeting, not every year, but we come occasionally, but we don't often come in a speaking role. We're often here looking at what kinds of new things are emerging from space biology in particular that might be relevant to our program. Today we have 4 speakers. I'll be, as Jennifer said, I'll be leading off talking about the what, where, and why of the Human Research Program. And then I have 3 of my colleagues from the Human Research Program who will talk about new kind of deep dives on brand new projects that we're standing up that I think might have a lot of relevance to the area, that the space between what space biology is usually doing and what HRP is usually doing. So I'm hoping that you'll find it all interesting and it might be stimulating for you to come and work with us at some time in the future. So I'll introduce briefly the other speakers. First is Dr. Jen Fogarty, and Dr. Fogarty is the chief scientist for HRP. I'm not going to go through their bios; they're on the website, just to save some time. And Dr. Fogarty is going to be talking about some work we're doing in precision health these days. After Jen will be Dr. Steve Platts. Steve is our deputy chief scientist at HRP, and he's going to talk about a systems biology project that is coming up, and it's actually done in tandem with the space biology program. And then finally, we have Dr. Robin Elgart, and Robin is the element scientist for our radiation research group, and she is going to talk about a translational radiation research project that's just becoming that's just coming up to speed in our program. So hopefully this will all be interesting to you and will drive some questions. I think we're going to wait until the end after everybody has spoken before we have questions for today. So I'm ready to go, Jen, whenever you're ready. So as I mentioned at the beginning, I'll be giving kind of the what, where, and why for HRP. I think most of you have at least some familiarity with HRP, and hopefully we'll bring you up to speed on what's new and what things we're doing and how we're doing them and where we're doing them. So next chart, please. It's really an exciting time to be at NASA, especially in HEO these days. We are working tirelessly to start setting up a set of new programs that will bring us back to the moon for the first time in more than 50 or almost 50 years. Moreover, more importantly, it will also set up our capabilities to start going to Mars. HRP is really focused on trying to reduce risks for crew members on exploration missions. And the target for us, the primary target for us has been Mars. That's the farthest destination that we were thinking currently that we might be sending crew members. So that's where we're going to head out. So the next chart, please. The way we do our work really starts with trying to understand what crew members are going to be exposed to during deep space missions. I'm sorry, could you switch back to the last chart, please? Yes. And I'm not sure which one is showing. It's the one that's on the left that I see. Is that correct? Okay, good, thank you. So, our mission, as I started to mention, is to do the research that underlies the risks that crew members might experience during deep space missions. And we do that through a pretty robust program of basic, applied, and operational research. And the outcomes of the research, like all research programs, start with knowledge. So we deliver knowledge back to the agency and back to the medical folks within the agency that's relevant to crew health and performance. But unlike other research programs, we also have some more operational deliverables. We deliver the basis for new health performance and habitability standards that the agency will use on these missions. We build risk mitigation solutions for those risks that might put the crew members at risk of not being able to complete their mission or having in-flight or long-term health concerns. We have to develop mitigation solutions, which include countermeasures. And also we develop medical support technologies and habitability technologies that we deliver to the programs. So we're not exactly a discovery research program, we're more of an operational research program. Next chart. The way we choose to prioritize our work starts with understanding what the stressors are to humans in all space missions, but in particular in deep space missions. And we've kind of parsed that down to 5 roughly orthogonal stressors: altered gravity fields, hostile closed environment, radiation, isolation and confinement, and distance from Earth. And like we like to do at NASA, we've color-coded these using a sort of a stoplight chart. You'll see nothing is green. This is for a Mars mission. But the 2 at the top, altered gravity and hostile closed environment, we have more than 50 years experience in low Earth orbit about how to deal with these problems for crew members. And so we have some insight into what we might expect in very long duration missions. Radiation, we also have some experience with from exposures in LEO and from the brief exposures crew members had during the Apollo mission. But we still know quite a bit about radiation. It gets much worse when you get beyond the Van Allen belts because of the fluence of galactic cosmic rays. And so we cover that as orange. But we have very little understanding of isolation and confinement, distance from Earth, because we just haven't experienced those things. So those right now in our minds are red hazards. We go to the next chart. When you think about the impacts of these hazards on human beings, you need to start thinking about how they might interact with one another and what systems they might affect. And I did steal this chart from Julie Robinson, who developed it for another presentation about a year ago. But if you think about the effects that might interfere with human performance for successful mission operations, they include physiological effects, psychological effects, and cognitive effects. And if you look at the impacts of the various stressors over on the left, you'll see that all of them have an effect on multiple aspects of the performance, except our altered gravity. Altered gravity is predominantly affecting the physiological systems and causing changes there. Now there is some vestibular disturbance that can cause some effects in cognition, at least transient, but for the most part, it's a single system impact. The rest of them all have multiple system impacts. Next chart. To really do our work, though, we needed to pair up with the Health and Medical Technical Authority from NASA, the docs who have to take care of crew members, and to try and look more systematically at all the risks that crew members might be exposed to. This chart, which I don't expect you to be able to read, is a list of all the risks that we're working on today. They're segregated in the left column by hazard, so you get an idea of how many risks are associated primarily with one hazard or another. But what I really want to show you is as we go across to the chart from left to right, we start seeing some different design reference missions. And the first 2 columns are missions that would occur in LEO, and one is for less than 30 days, and the other one is up to a year, the one on the right. And you see that most of the risks are colored yellow or green for those missions. Those are ones that are captured by ISS today and by whatever commercial alternatives to IRS or ISS might be there in the future. All the way on the far right, you see a Mars mission, a 1 to 3 year Mars mission, and you can see that there are quite a few red risks. Those are the ones that are the highest priority for us. Red in this context means that there's really a high likelihood and/or a high consequence with that risk to the crew member, either health or performance during the mission or long-term health after the mission. And so it's really something we don't want to fly any crew members if they're going to be in the red risk category. Yellows, we may or may not be able to fly crew members. They're kind of moderate likelihood or consequence. And so almost all of them are going to require some new standards or some countermeasures. In between, we have basically what we can learn from Artemis. We have lunar orbital and lunar orbital plus surface. And then the fifth column, the one just before the Deep Mars mission, is a Mars shakedown cruise that we're expecting to do sometime probably in the early to middle part of the next decade in cislunar space with a full up test of crew members aboard, probably 4 to 6 crew members aboard this exploration habitat in cislunar space with all the countermeasures and all the time delays and everything built in for a full 1 year simulation. So we're kind of be moving towards that to make sure that we're ready to go to Mars. Next chart. So where we do our work is shown in this chart, and how we're strategizing the work we do to make sure we're ready for Mars missions when they come up. And this is an uncalibrated timeline with time moving to the right. Our primary location for research is on the ground. That's the lowest bar here, ground analog research and laboratory research. This is where most of our work is taking place. The next step up is to go to LEO, low Earth orbit, and we of course use ISS pretty heavily these days, and we will be as long as it's there. But we know that there's going to be some transitions to future platforms. We expect that there's going to be a lot of questions that we can still answer in LEO that won't require us to go into deep space. But we're now also starting to enter the era of cislunar and lunar surface work that you can see in the light browns triangular section there. And we are already starting to work on planning what we'll be doing in cislunar space and on the lunar surface as we move forward. And then finally, we have, before we would go to Mars, we have this high fidelity test mission that I mentioned on the last chart. A lot of the work that we're doing today is still risk mitigation research. A lot of it is kind of basic and understanding mechanism testing countermeasures. We're also starting to overlap with systems testing and validation where we think we have countermeasures or approaches. We're starting to do those in LEO, and we'll continue doing both of those kinds of work in cislunar and on the lunar surface as we move forward. So next chart. So you might wonder how we simulate exploration stressors on Earth, and these are the facilities that we make available to all of our researchers to be able to do that work. In the upper left-hand corner is a radiation facility. This is sponsored by the Department of Energy. It's at Brookhaven National Laboratory on Long Island, and it's been in operation for more than 15 years now. I think 17 or 18 years, if I remember correctly. In that facility, we can simulate the full spectrum of galactic cosmic rays now in sequence, so we can do them all at about the same time, or close enough to the same time to think of it that way. The only downside to the facility, from my perspective, is that it's unethical for us to put humans in front of it. So we're limited to rodent species for the most part. We may move to mini pigs or some other species as we move forward, but it still requires us to do some kind of translation for what we see in the model organisms and how to translate those up to humans for the future. Below that, we simulate artificial gravity and have for 3 decades or more using head down tilt bed rest, which causes the fluid shifts and a lot of the other muscle and bone wasting that we see in crew members exposed to long-term microgravity. And we've been using a facility for the past few years that was built by the DLR Institute for Aviation Medicine in Cologne. And that facility is probably the world's finest. It's the newest and was designed exactly for being a ground analog. They can bring a lot of throughput of crew members or of test subjects for us, human test subjects for artificial gravity work. But they also have a capability to superimpose part of the hostile closed environment. They can change the CO2 concentration of the environment and make it more like what we might find on the ISS or in other space vehicles going forward. So we can look at two stressors at the same time there. The middle column shows how we do isolation and confinement as well as distance from Earth. And we have two facilities we use for that. The one at the top is the HERA facility, and that's at Johnson Space Center. And in that facility, we run campaigns once a year. We have four missions within a campaign with 4 subjects each that stay inside of that chamber for 45 days. And down below, we have the Russian facility at IBMP, the NEC facility that we've been using in cooperation with them. And in this facility, we're doing multinational, multicultural studies with 4 to 6, usually 6 crew members, but they stay for a much longer time. So we did, a couple of years ago, we did a sort of a shakedown test with that facility. That was a 17-day campaign, and then we moved to a 4-month campaign. In this spring, we were supposed to start an 8-month campaign, and unfortunately, the coronavirus pandemic has gotten in the way of that. It's been slipped until probably the fall for a start time for the 8-month, and then the year after that, we're going to do 1-year campaigns. For anybody who's interested in volunteering to be in isolation and confinement and are astronaut-like people, we always have notices out for test subjects for either of those 2 facilities. In the upper right, we sometimes still use altered gravity testing aboard parabolic flight. These are for the rapid time constant systems in general, so looking at vestibular effects and cardiovascular effects predominantly. And then finally, we have Antarctic winter over capabilities we've had for the last 3 or 4 years, if I remember correctly. And these are provided by the National Science Foundation. We get to do studies on people who are going down to winter over in Antarctica. And for the most part, we've been using the South Pole Station, which is in the middle of that Antarctic mass there. We've also done a lot at McMurdo, which is south of that or below that on this picture. And then this year we started doing some work at a more isolated station with fewer people, way out at the end of that 10 o'clock peninsula called Palmer Station. So we have regular campaigns for all of these that we set up, and we organize all the complements of studies that go into these things. And we're usually doing calls that will allow people to be able to propose to do work that is relevant to our mission in these facilities. Next chart. Of course, you're all very familiar with ISS. We've been using ISS for many years since it began. We use the crew members as our test subjects, also as test operators, and we basically do studies on every crew member who flies and every USOS crew member who flies, I should say. We don't often do stuff on the Russian crew members. We typically have somewhere on the order of 250 hours of crew time per increment pair, something in that order. Lately, it's fallen off a bit because of all the changes, but we have a lot of help from the ISS program office in trying to prioritize the work we do because of its relevance to the future exploration missions that are coming. Next chart. The next step is to go to cislunar space and then to the lunar surface. And there we've already started work with the Gateway folks about how to do some studies on the initial phases of Gateway. And in fact, we've been leading, Lori Abity from HRP has been leading an Isselzweig Tiger team that was tasked with developing an integrated set of human research payloads for these early capabilities. those things that we ended up down selecting to 11 studies, I think, and about half of them are sponsored by the US, but all of them have multiple international partners associated with them. And they're undergoing evaluation now by the programs to see what we can and what we can't do. But we're already starting to use these facilities to answer the questions that we need to answer. Next chart. You may or may not be aware that we have an external partner called the Translational Research Institute for Space Health, or TRISH for short. They've been in business for about 4 years. And we put them together to be an external skunk works to look at what's going on in terrestrial biomedical research and technology development, and things that we might not see from our low knothole on the fence, and to be able to bring those things into the risk mitigation strategies for us. So they've been making a lot of progress over the first 4 years that they've been in business, and they're starting to bring a lot of deliverables to us now in the past 6 months or so. The stuff down below I pulled from their website, and the link below gives you a way to go and look at what's going on over at TRISH at any particular point in time. And the next chart. As I mentioned up front, we have a very synergistic relationship with BPS. We see space biology as an important partner to us in many areas: microbiome, plants, gravity as a continuum, gene lab as a repository, chip tissue chip validation, pharmaceutical testing, and others. Together, we share investigators, partners, and platforms. And despite the changes in organizational structure, where we're a little bit more separated, we're all close friends and we all integrate together. I'm talking with Craig and Nikki on a regular basis, and we're starting to work with Sharmila now as well through the Chief Scientist Office. So it's an important relationship, and I think it's really very useful to us as we go forward to get some of the basic understanding that can be translated up to our risk mitigation research. And then for the last chart. And the last chart is really an advertisement for you to get involved with our mission. In this year, we're funding currently 224 research grants to 182 PIs at 106 institutions in 26 states. We are very open to good ideas from wherever they come from, and how they fit into our program. And we're very open to having new people come in. We have some sort of regular flagship solicitations that go out about 3 times a year. And attached to those are something we call omnibus, which are either for unique ideas or in some cases for investigators new to HRP. In other cases, they're to test out a protocol and get preliminary data to write a flagship proposal for the next year. Our opportunities are listed here. Most of you are aware of NSPIRES, and that's where our opportunities come out. I've also listed where the Translational Institute has their solicitations, and they put out periodic solicitations, sometimes 3 or 4 times a year as well, usually for a little smaller group of people. So that's all that I have to say, and that's an introduction to pretty much the what, why, and where HRP does its work. And I think we're going to move on now to the next speaker, who will be Dr. Jen Fogarty, and she's going to be talking about a precision health project that we have going on. So thank you very much.
Thank you, Bill. I appreciate it. The introduction, I think it helps people understand who HRP is if they didn't know, and the scope of our work and some of the structure that helps inform how we build our portfolios. In my title slide, I want to focus on the subtitle for a second, Precision Health, Reducing Risk and Resources for Exploration Missions. This is one of the elements I think that potentially gets overlooked when we're doing development and research, is that the constraints of the missions are quite severe. We will not have the mass, power, volume, not even what's available to us on ISS. The projections are pretty meager. So we continually have to work at becoming much more efficient and precise about what we do. So we go to the next slide. So when I start my talks, I really want to focus on kind of where we come from, both as a species and culturally, what we're used to having and how we're used to having it. So when we look at Earth from far away, essentially taken from the surface of the moon, it gives us a vantage point of kind of the richness and the texture available on the surface. The sights, the smells, the sounds are all very intrinsic to who we are as humans and what we experience regularly. And those won't be available unless we recreate it in some way, unless we find a way to give stimulation to people that mimics that and provides them what they need. The other aspects are the easiness with which we get resources to a large extent. Going to your refrigerator, going to the store, stopping to get items, resupplying yourself regularly. If you don't like one choice you have, you can go get another. With the idea of exploration missions, you have to preconceive these things and pre-plan them, and even in a more extreme sense, pre-deploy them. And you really don't get second chances for things like a Mars mission where there's no return and resupply is conceptually not available because of the distances these vehicles will travel. A vehicle launched later can't catch the vehicle that's been launched. So there's some option once you're on the planetary surface to be sent resupplies, but it will be a while. So we go to the next slide. So when you compare the status of Earth and the texture, we go to a much more stark version of Mars. And we realized intellectually that it's not a habitable place for humans. We're going to have to create the entire environment for the people who will be sent there and sustain them for the period of time they're expected to be there. In addition, the transit vehicle, to help conjure up the complexity and the holistic nature of what it has to bring, if you use the word vehicle, it seems a bit more temporary, and we have vehicles at our disposal here, cars, planes, trains, but it's not like that kind of vehicle. It's more on the order of an exoplanet with a closed environmental control system, life support, all of your resources such as food, water, waste disposal, in addition to being an operating vehicle that can actually take you from point A to point B and do the mission objectives that it's designed for. So the austerity brings up a lot of those constraints that if you could be more precise about what you were planning for and be able to more confidently predict what may happen, could we be much more efficient at preparing, but also being mindful of some built-in agility that we'll talk a little bit later. So we can go to the next slide. So given the constraints and the austerity of the planet we're going to, why would we dare to do it? I know this question comes up regularly, not just to the life science people, to the astronauts, program managers, administrators. Why would we do such a thing? I think there's many reasons, and they usually can be very personal. But overarchingly, as a population, as a species, we definitely have a predisposition to explore, to understand what's beyond our current capabilities. I also think that exploration has brought capability to us that we could have never anticipated. Necessity is the mother of invention. If you have to do things you have never done before, you're going to have to figure out, and you will learn very hard lessons in the process. We work in a couple different spaces while we're trying to achieve these goals of putting human boot prints where only rover tracks exist from a human impact. And that's really about stretching the limits of how we successfully survive environments that are inhospitable. We learn a lot from the expeditions on Earth, whether they be to high peaks such as Everest or deep sea diving, mapping the surface of the planet. There are extraordinarily different pressures and environmental stressors that occur that drive humans to have to do things in a very different way than they do at relatively one atmosphere, sea level. So I think we've had to undertake a very concerted effort to look and think about the complexity and gather evidence about the complexity of the environment on the human and the human's ability not only to be healthy, but to perform and perform at the standard the mission requires. So if we go to the next slide. So when we think about the types of solutions we're going to need to develop, we come up with a bunch of characteristics that are commonly discussed to help people understand the pressures, the constraints of what are going to be brought to bear. So autonomous, what does that have to do with? Well, you can't be dependent on the ground. The communication lags that will occur are going to force operations that we don't currently execute today in many ways, because we have easier access to the ground in low Earth orbit that will not be available. Well, it'll be challenged in lunar orbit. There'll be longer delays. There could be blackout periods. On the Mars class missions, that will grow to up to 20 minutes each way. But also there's bandwidth issues, not unlike we're all experiencing at home regularly during remote work, that as much as you may want to attend or you may want to communicate, that system is disabled and it's not under your control. So we have lots of debates about what system might get what parts of the bandwidth to send packets of information back and forth. So we have to think a lot about allowing the crew to operate on their own without the expectation of ground support, because it will not be anywhere near the almost instantaneous ability to respond or be available to troubleshoot. And clearly, the vehicle itself can't rely on command and control from the ground. It would take way too long and be completely ineffective for what the crew is dealing with at times. Light, lean, that comes up to launch mats. It's very challenging to get items off the surface of the Earth and into space. It's very expensive to break the bounds of the binds of gravity. So we're constantly under pressure to make sure that whatever we're doing has been reduced to the lightest and the leanest form, but also robust. So, and which sometimes can be complicated to provide paradoxical characteristics. Is it's got to be able to last. Bill went over some of the hazards. I'll talk about them again in a second. That it's going to be put through its paces, whether it be a radiation environment, the fact that it has to be low noise because it has to interact with the crew 24/7. How much heat does it produce? How does it manage the heat it produces? So it's very complex to provide something that's also a robust solution. And on the human side, if it were biomarkers, robust meaning that it's repeatable, that we can trust the signal that we're measuring. It also needs to be on the order of some level of comprehensive. If we needed a solution, a point solution for every point problem, clearly going back to the launch mass and constraints, it's not going to match the scenario that we're in. We also look at solutions that are driven by data and evidence, and also help us move toward predictive analytics, that we have decision support. It doesn't decide for you, but it can amass data that any one human couldn't accomplish acquiring and synthesizing in a timely manner, but that we could provide that database behind the scenes, so when the crew asks a unique question or has a unique combination of events occur they need to respond to, that the intelligence of an entire community that was on the ground that is no longer readily available can be brought to bear. There's also aspects of agile and responsive to the changing environment. During the transit, there are aspects of steady state such as microgravity, unless we provide an alternate countermeasure like artificial gravity. But as you'll learn, that the space radiation environment changes continuously. It has a spectrum, but it isn't homogeneous. So different particles, different effects of those particles could be occurring over time, and we're trying to understand that complex environment. The environmental control and life support could have varying capability over time. And then even in the fact that during the transit, the microgravity might be a continuous factor, we change environments incredibly quickly from microgravity, and in the scenario of ISS, we send you to 1G when we bring you back to Earth. The anticipation is to take you to a partial gravity field, 3H-G. So the more we can understand about an individual, their baseline and how they respond to provoked scenarios, we might be better off to be able to predict the outcomes that are going to occur. And this is where it starts to impact the medical system, because it's when you can connect the idea of changes of a group of individuals very precisely under the various stressors that we know will occur, and over this time course, how that may emerge to prepare a medical system, a performance system. We actually shifting into a crew health and performance system, because these need to be functional, not just healthy, but functional individuals to complete the mission objectives, that the precision will allow us the lightness, the leanness, the agility, and also the data to support that we believe these are the things that we needed to target and protect for. So we go to the next slide. So going back to the hazards and where we focus, and Bill had a slightly different colored chart. They're emerging over time. I think his was a little bit more up to date. So we definitely used a stoplight chart you saw on the risk profile that, it's very intuitive. Green means go. We know enough about it, and we understand how to mitigate it well enough, and we have what are called requirements we can deliver to vehicle designers and program managers to do that work. When we're in yellow, it either means we don't know enough about how to mitigate it, or we have unsatisfactory mitigation capabilities, or the fact that they cannot be integrated into a vehicle is just too large, too complex. It doesn't meet the other standards. They're in conflict with each other. The red ones could be red for lots of reasons. Instead of just being yellow and caution, red means all stop. That at this point, we've got big enough problems to solve that we would not be comfortable as an entity saying you're ready to go, and this is where we want to put our effort. So we spend a lot of time on the portfolios for radiation. I think Bill showed it as orange because it's in the space of the more we characterize radiobiology and understand its impacts on the acute phase during an in-mission versus long-term health, and our strategies to address those impacts could be very different and put us in a place where we can manage the risk, but it has to be transparent and it has to be accepted by the agency in terms of how that's going to look. One of the other aspects of spaceflight is you're going to have to accept some change. It's knowing what that change means in the human that really helps us to understand if there's negative consequences near term and long term. Clearly, isolation confinement weighed pretty heavily on the psychosocial issues. These are going to be incredibly challenging missions that we really have no experience base to lean on. They are stretching our knowledge base to an extreme where we're probably outside our comfortable limits of saying we can extrapolate from existing mission. And so Bill mentioned some of our analogs where we look at driving the issue and having isolation confinement for much longer periods of time to really help us understand where the vulnerabilities are and what countermeasures might be very effective. And part of the precision medicine approach would say, well, who's vulnerable to what aspect? And tease out if we can get ahead of it and do a lot more prevention rather than treatment after. And of course, distance from Earth has a lot to do with, one, the communication delay, but it also has to do with not being able to return. We often use that as one of our mitigation strategies, and that quickly does not become possible on the Mars scenario. So a couple of years ago, if we go to the next slide, NASA, under the leadership of Dr. Pulaski, Dr. John Charles, Dr. Craig Kundreit, had put together an effort. We were advancing the idea of long duration missions on ISS, which Dr. Pulaski mentioned, to use that long duration microgravity as a strategy in low Earth orbit where we have a lot of testing capability. We return the crew to Earth. But we also had the opportunity when Scott Kelly was picked as the one-year crew member to understand very poignantly on a molecular level what was going on with him against, expression of outcomes. But we also had his identical twin, who was a former astronaut, Mark Kelly, and to look at the idea of one person in space over a period of time, one person on Earth with a very similar genetic code, identical as they would say, but experiencing very different stresses over time. So how does the expression look and how does the function look? So they went under the same tests over the course of 3 years, really. 1 year before, the year during the mission in spaceflight, and 1 year after to understand a recovery. There's been some significant papers published talking about the gene expression changes and some interpretation, which is really challenging in an N of 2, but really an N of 1 if you focus on Scott Kelly alone in terms of his experience in spaceflight. And the difference was that Mark Kelly on ground did have some challenges going on in his life, and you would see some small subtle changes, but Scott Kelly clearly had some magnitude of changes that were impressive, but not all that surprising given what spaceflight is, the challenges in and around it. But it also really helped the agency establish policy and the culture of doing this type of work. It's really important to feel confident and comfortable and that we are meeting all of not only the law, but earning the trust, and we have the scientific credibility to do work that does genotyping, that does epigenetics. These are very personal attributes, and they're not only personal to the individual we're testing, but their family members. So there was a lot of infrastructure that had to be put in place so everyone was reassured that the science would be of value, but also that we could protect the integrity of the data and the integrity of the interpretation of the data. Because there are always areas, and as you know, with this being very predominant in the popular media, that when a paper gets published and a title of a paper gets out, it can be very sensationalized and people are off jumping to conclusions about the implications of a certain genetic code, when over time it starts to show you that it isn't that gene alone, or it isn't 100% assured that that gene will result in that outcome. That's actually the exception and not the rule. The genetics are incredibly complex, gene on gene, environment on gene on gene. We have a lot to learn about how the body regulates its response using genomics, which spans from DNA all the way up through functional proteins and the expression of all of those along the way to result in how the human functions. So if we go to the next slide. So taking what we learned, and we are going to continue to do long duration studies that span the spectrum of molecular through functional, but we also wanted to very deliberately look at individuals and create a resource where data analytics was the priority. And when you look at the literature around precision health and deep phenotyping, and of course we cite here a Price et al. Paper, which is Lee Hood is the senior author and has world renowned for his work in deep phenotyping of individuals. And it's a very elegant way of trying to map not only genetic code, but gene expression to environmental stressors. But it is also very dependent on the collection of a lot of metadata about the individual and the environment they're living in throughout time to understand the relationship between who they are fundamentally on the molecular level and how does that look on the other side. And instead of just correlations, is there a sense of causality where we would have a lot of confidence not only to measure the change, but then to act upon it. In parallel, you also want to develop something that can allow you that action. Because I think in Dr. Pulaski's presentation, our goal as an applied research program is to deliver results to operations. And in order to give the clinical world confidence, that they can do something with the data, we have to make sure that we're actually developing that solution while we're uncovering the problem or the relationship. So the Precision Health Initiative is really designed to have HRP do the lion's share of the data collection using gold standard methodologies, but then open up the competition for the analysis side. And what I think is interesting, we had a great presentation by Gene Lab from Ames Research Center recently, Dr. Sylvain Costas, and they are way in front of us with the animal data. As you might imagine, there aren't as many barriers ethically to the animal data that we run into with the human data, and they have been approaching this and archiving the data in a way that really facilitates the concept of deep phenotyping and precision research on mammalian rodents. And then we can also help understand the translatability, which I think Dr. Platts might speak to with the systems biology overview. So if we go one more slide. So there are elements of the deep phenotyping and the precision health that'll feed into the precision health initiative that we do today. One of them is HRP spaceflight standard measures. That is more on the physiological and functional end. We have quite the portfolio sitting out there, and we actually very publicly list the measurements we take and make them available through the Life Sciences Data Archive for request. We are transitioning to what's called the Spaceflight Omics Archive. We currently have an effort called Repository, where we collect blood and urine off of crew members pre, in, and post-flight. We do a lot of efficiencies in terms of we don't take them as a primary function, but always secondary to all the sciences going on, because we have a very large science queue to continue to facilitate The Omics Archive essentially takes repository and enables it to allow genomic research. So the types of tubes have been changed, the way we collect samples have changed, and the specific collection of samples such as buccal swabs and fecal samples are all geared toward making sure that we have samples that enable this process, but doesn't force every individual PI to come forward and get the collection done. We do the collection, we have both data generated, and we have sample archived. We're moving forward with space medicine on a pharmacogenomics effort. This is at the clinical level today. You can get pharmacogenomics done to help you understand your cytochrome P450 polymorphisms and understand how that ranks against drugs and how you metabolize drugs. And it is something that I think would be incredible metadata for the researchers to have throughout the process, not only what drugs people might have been taking while they were before, during, and after spaceflight, but how they metabolize them. And it could be part of interventional studies as well. To the right side, data integration analysis, we're still kind of formulating and evolving our data management capabilities, both the lifetime surveillance of astronaut health and the life science data archive. It's incredibly important to us that we have robust archives that actually not only store data, but we can redisseminate and work with our epidemiologists and our archivists. So an integrated hazard response is also a way to show gene expression against outcomes. And a lot of that comes from terrestrial data in terms of all the robust work that's going on from NIH funding. And we leverage heavily off of that because in the end, these humans are still humans going in spaceflight. The environment might be unique, but potentially the responses are well in a line that you might see under other stressful conditions, which is the concept of using ground analogs to provoke one or more of the hazards that we can replicate versus the complex environment of spaceflight. And the last one is still in work, and it's called molecular dosimetry. That is a nod toward radiation, but actually it's not exclusively thought of as a radiation effort. The concept of spaceflight and all of the hazards that the crew members are exposed to put in its totality environmental pressure, and that will produce potentially unique gene expression signatures that we need to understand. So that's part of the archive, and that's an evolving piece of work that you'll probably see come out in some of our solicitation topics over the next year or two. So we go to the next slide. So the advantages of precision health approach for exploration are really meant to be proactive health and performance risk management. Instead of waiting for something to appear and being responsive to it, we'd like to understand where the vulnerabilities are and be able to predict where we think things might be emerging so we can actually do surveillance on them. So the idea of identifying things on the genomic and molecular or the molecular level as biomarkers and identify them as risk factors, and then establish baselines and use surveillance pre and post mission to understand how those biomarkers change, but when does someone reach an actionable level and what action should be taken. We also have to use that then to understand how to do in situ analysis to measure the marker and have effective and efficient tools, because we also had to pre-predict what the action was. So they're very much interlinked. We believe we're going to reduce resources by providing this custom approach. The idea immediately translates to pharmaceuticals and how we pack the medical kits for a crew on an exploration mission. It's a little different than ISS, where you have turnover of crew and you need a more generic pharmacy for all the people that might be going. This is going to be a very precise endeavor for something like the first Mars mission. But it could even be effective on something like exercise protocols, how someone uses resistance and cardiovascular exercise, how much stimulus does any one person's musculoskeletal system need to stay as robust as we need it for the mission and to maintain their long health. If we go one more slide. There still are going to be really some large challenges, and this is where we're going to continue to interact with the academic and research communities. The amount of evidence, the amount of data to drive this is not insignificant, as you know. There are many global efforts, the UK, NIH, collecting large data sets, people being asked to sign up. I believe NIH is called All of Us, not only to donate your medical data, but genetic sequencing, so they can map over time and start to understand how relevant it is to do precision versus when does epidemiology make sense. And we also, the challenges of reducing resources mean that you're going to have to screen a lot of capabilities before you can down select what needs to go on an exploration mission. And then inevitably, we're going to have to come to the table to do what we call is risk trade. We need quantifiable data to sit at a table and understand when you're choosing against how much water is brought, how much medicine is brought, how much hardware is brought to support all the other science that needs to go on. Those are real discussions. And the more accurate and precise and quantitative we can be, I think the more successful the entire mission can be. So I really appreciate the opportunity to be here today. I appreciate Bill for giving me a lot of latitude in the program to push areas that in the past we were kind of concerned about or weren't mature enough. So he definitely enables me to reach out to as far audience as possible. So I very much appreciate it.
Okay, thank you. Sorry, I had some problem with my mute button. I wanted to introduce Dr. Steve Platts, who's the Deputy Chief Scientist for HRP, and he's going to talk about a systems biology translational project that we have that we're starting to ramp up now.
Thanks, Bill. Next slide, please. So what I'm going to talk to you about today is the evolution of a new project that we're starting in HRP. And just to give a little background, I wanted to talk about the way we do our science work within HRP. So if you look at this figure on the top left-hand side, we look at our evidence base. As Bill mentioned, we are an applied research program. We're concerned with risks that are identified, and we try and mitigate those risks. And so we have to identify the evidence that leads to those risks, and whether that evidence is clinical, operational experience, or from research experiments that have happened in previous programs like Shuttle or Apollo, and those generate that stoplight chart that you saw earlier. But then we go a step further and break that down into the knowledge gaps that then contribute to those risks. From those gaps, we develop strategic plans, schedules, and we figure out when and what we need to do for research. So that leads to solicitations, it leads to directed research, all of which goes through rigorous peer review, very similar to the NIH peer review level. And then we wait for the results. And so we get these data back from our investigators, and we look at those data and we analyze them, and we decide what we need to do next. Do we need to modify that risk? Does the data help us to mitigate that risk, or do we need more experiments? So it's this cycle. Then that feeds into that new evidence base. So we end up with this cyclical process. And I want you to remember that cyclical process because that applies as we go forward and consider systems biology. Next slide, please. So we're standing up this new project, and we call it, Systems Biology Translational Project. And so what does that mean? If you talk to 100 different researchers in the biological fields and you ask them what systems biology is, you'll get 75 different answers or something resembling that. It means a little bit different things to each people. And so this is how we're defining it. So it is the integrated use of genomics, computational modeling, and wet laboratory data collection and analysis in a cyclical fashion. So it feeds into each other. It's constantly evolving. We don't have stovepipes or stopgaps where we end up with dead ends or things stop. And so we have done an excellent job in the past of doing the functional and performance testing. We've been doing also a fair amount of computational modeling, but rarely are they prospectively integrated into single experiments or single projects. They tend to be, this study is over here and they're looking at a physiological issue, they generate data, and then we have another solicitation over here that takes those data and does modeling on it. what this project is going to do is look at all three in an integrated and prospective manner so that we can more thoroughly investigate our risks and then hopefully more quickly and more cost effectively mitigate those risks. Historically, the thing that's been missing from this triangle has been genomics, and Jen gave you a little bit of a primer on twins and how that helped us. But if we go to the next slide, omics was really the missing link. And so until we got to twins, we did very little omics work. Really, all that was done was some metabolomics and some SNPs and things like that, very little omics work. And you have to understand that the astronauts are a vulnerable and protected population. They are celebrities, if you will. They have people who search them online. They have people who follow their careers very closely. And so when we do genetic analysis or genomic analysis, it's very, very important, as was mentioned earlier by Jen, that we have all of the correct protections in place so that information doesn't get out that would jeopardize the privacy of the astronauts. And I think the twin study really helped us show that to the population. Next slide, please. And so the introduction paper was published in Science magazine, and we had a lot of firsts with the twin study. So we had 10 different research teams participating. We had over 80 individual investigators participate on one set of twins. They went between 8 states, so we actually had to follow one of the twins around, and we did data collections all over the country. Lots of blood sampling, lots of other data collections, 340 days in space. So some of you may have seen um, Commander Kelly had a picture on Twitter of him holding up a sign that said 340 days close enough for government work, because we call it the 1-year mission. But you know, it was pretty good. That's pretty good for us, 340 days. And so lots of firsts that happened here. And again, the one that I want to point out is that first foray into the omics field in a serious and systematic way. Next slide. And this is a great summary figure that our communications team came up with, and I absolutely love this one. I love to show it. And it shows the 10 different studies that we did. So we did biochemistry, we did metabolomics, we had a microbiome group, we did proteomics, we had one group looking at telomeres that showed some really fascinating results, something we never would have predicted from that telomere study. Integrative omics, we did cognition, gene expression, epigenetics, and the immunome. So lots of different things, lots of data, tons and tons of data, unbelievable. But by doing this in such an integrated way, we showed that the systems biology approach can and would work. And so this new project is going to incorporate that knowledge, incorporate what we learned to move forward and get a nice integrated product. Next slide. So the objectives, the goals of this project are to utilize all of those things that I described earlier to look at multi-level, and this is important, going from the cell to the tissue to the whole organism, but also multi-system, whether it's cardiovascular, renal, neural, immune. And an example here, since I'm a cardiovascular physiologist, and it's one that I know and is dear to my heart, if you just look at the cardiovascular system and you're studying, for example, blood pressure control, but you're ignoring the renal system, which is the long-term control mechanism for blood pressure, and you're ignoring the neural system, which controls the very short-term blood pressure control systems, and then you're not looking at endocrine systems, which controls all the intermediates with angiotensin 2 and aldosterone and all those things, you are missing the boat. It is not one system. So we are looking multi-system. And then finally, we're looking multi-species. So we're not just looking at humans. We're not just looking at rodents. We're trying to figure out how that translation occurs. And that starts by working with our friends in space biology, looking at not only their genomic data that they've gathered on rodents, but also on the research data from more physiological, more functional experiments, tying those together and seeing how we can link those to the human. Next slide. And so in order to get this project moving and to generate a proof of concept, we're looking at two things. We're taking a two-pronged approach. One is we're starting from the ground up on a cross-cutting and integrated problem, and that's inflammation. So I think a lot of people here can recognize that inflammation is important in our medical health here on Earth, and we believe also in flight. And so we have experiments going on now between the different elements, looking at inflammation in the CNS, inflammation in the cardiovascular system, inflammation as it relates to immune. What we're going to do here is pull that all together and then get the modeling going, get the omics going, look at it from that view and get a bigger, more holistic view of the problem and then take it from there. Our second approach is to take a project that's already in motion and try and apply the systems biology theory to that project. And we've picked SANS. And most of you know that SANS is a project that's been going for quite some time. They're looking at how spaceflight affects the brain and the eye and things like that. And they've done a great job of looking at the physiology and the pathophysiology, and there's been a little bit of omics work, but not very much. We're going to bring that to the table. There's been a little bit of computational modeling, but not very much. They haven't been tied together. It hasn't been integrated. So our second approach is taking the SANS project, adding these other components to it, and trying to bring more value to that project. Next slide, please. And as I mentioned, we have quite a few space biology collaborations built into this project. So in order to fully integrate the genomics, we have to start with the animal models, and GeneLab has those data in there. And we will be working with space biology on how to mine those data, generate heat maps, and then try and apply that translationally to the human. And then finally, creating useful and applicable computational models that relate both to the genomic data and the physiological outcomes, so that then when we generate those data and we can apply those models to them, we can change the models if we need to, discover new hypotheses on things we should test. It will be that cyclical process again, coming back onto itself constantly to improve with every iteration. Next slide. And so this is kind of a NASA geek kind of a figure, but I really like it. So we worked on these new strategic plans for each risk, and this is the strategic plan that we came up with for this new project. And you can see on the left, we start with data mining. What do we have to do for that? So collecting genomic data, mining both the animal and the human, holding a workshop with experts in the field to make sure that we're going down the right path. And then we're even identifying some deliverables down there on the bottom. What data do we need to use for modeling? And then the middle here is mapping and modeling. That's getting going, actually starting to do the work, generating a network map of the relevant inflammation pathways, doing the same thing for SANS. Getting those things going. And then finally, once we collect those data and we do the analysis, how do we test and validate? So using those data to leverage and the models, and then generating those results, validating those models. That's going to be an important thing for us. We're big on validation. We don't like to just generate models and then use them. We have a very strict validation protocol for doing that. And then hopefully our end goal is being able to make recommendations for countermeasures. Next slide. And so we've identified a few things that we would consider success criteria. So we want to get enough data so we can generate a good and useful network map and computational models. My particular goal is hopefully those maps will point us towards some potential common pathways that we can then take advantage of in the inflammatory process. And then we came up with this number of 70% confidence in our models. We think that that number is sufficient to make the model successful in allowing us to predict what's going to happen and then to test further hypotheses. And so given that those we predict, that we'll be able to complete this in a number of years, but the bigger picture really is not just to limit our systems biology approach to this one little project in inflammation in sands. In the big picture, we want to be able to apply this to all of our research projects. And hopefully, really over time, this project will put itself out of business, because what we'll do is we'll show that this approach is useful, it will save us time, it will give us a better result, and then slowly over time, this approach will be intercalated into all of our other projects. And then when we say systems biology, instead of thinking, oh, that's a new project, we'll think, of course, that's just the way we do business. So that's our thinking for the long term. And I thank you very much for your attention. And that's all I have for now. Thank you.
Okay, thank you very much, Steve. And we're now going to move to the last speaker, Dr. Robin Ellegart. Robin is the newest member of our team. She's only been with us for a few months, and she's coming up to speed, but she has been working diligently since she came aboard on developing translational radiation research and countermeasures projects. So she's going to tell you a little bit about that today.
All right, thank you so much, Bill. And first I wanted to say good morning, good afternoon, good evening, wherever you are. I'm the space radiation element scientist. I started back in April, so I just about got my legs under me, I think. So go ahead with next slide, and we'll jump into this. All right, so I wanted to introduce you to our new team. We've had a lot of changeover in the space radiation element. So here's the main leadership team. And of course, the goal of the space radiation element is to ensure that crew members can safely live and work in space without exceeding our acceptable radiation health risks. And so on my team, I've got a fantastic deputy element scientist, Janice Sawaski, and I have 3 fantastic discipline leads, Greg Nelson, who covers CNS, all the brain stuff, both early and late, Brock Sykes, our cancer discipline lead, and JP Saha, who is in charge of our degenerative tissues. And on our management team, we have, these are really the people that keep us going. This is Jason Weeks, our element manager, Nick Meyer, who is our deputy element manager, and we have a fantastic science integrator, Lisa Stevenson. And I do want to highlight, we have a element inbox that will go to our team and get you routed to the right place. So feel free to email us at any time using that email. We don't promise to get back to you within minutes or hours or even days, but if you don't hear from us in a couple of weeks, just give us another ping. We won't be offended. Next slide, please. So I just want to give everyone the briefest of brief little background on space radiation, because it's a complex topic. There's people that have been in this field for 10, 15 years, including myself, that it's complicated and you're learning things every day. So the space radiation environment is the majority that we are concerned about are high energy particles. Now, these are fully charged atomic nuclei, so they're physical particles zipping through space. They deposit energy along tracks. So you can see in this sort of artist rendering on the right here, that's what a charged particle track sort of looks like. It deposits energy sort of as little explosions along its track, compared to, say, a gamma radiation source, which is more random in its distribution. So if you do one click, I'll see if this works. Awesome. So I like to think of high energy particles more like this pug in a tank, compared to our lower LET or linear energy transfer radiation as more of this Sharpay with these little cute Cupid arrows that might bink, 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 bink all over the place. But the pug in the tank, you know, that's getting launched and that's going to go straight through some stuff and really do some damage along the way. Another thing about high energy particles is they actually travel along magnetic field lines. That does become important when we talk about solar particle events, as well as our magnetic field. And these are really difficult to shield. If you can imagine sort of a wall of ping pong balls, that's what any sort of material is made up of, right? And if you take a baseball and you throw it through that wall of ping pong balls, what do you have on the other side? You now have a baseball flying at your head, but you also have ping pong balls flying at your head as well. So that's what is known as secondary radiation. So it tends to, these high energy particles tend to be very difficult to shield, depending on their energy. And what we're finding is that these are also more biologically damaging. As you can imagine, like our pug in a tank, having a missile blown at you is probably going to be a little bit more damaging. The kind of damage might be a little bit more complex than just, getting an arrow in your shoulder or something like that. The composition of that radiation out in space and the energies really depend on the source. And we have three main sources. I won't go over them in any detail. Just know that we have trapped radiation. These are known as the Van Allen belts. This radiation is trapped within our magnetosphere. That's coming from the outside. And then we have galactic cosmic rays. This is the one that people are very familiar with by now, I think. And then we have solar events. These are emanating from our specific sun when the sun decides that it's sort of had enough. Its magnetic fields get a little angsty. They say, I'm done. They release a bunch of energy. And sometimes we can get particles that come with us with that. All right, next slide, please. So now you know everything you need to know about space radiation. So what are some of the challenges that we have in studying space radiation in terms of human health? All right, so we have a number of challenges, but I'll highlight a few of the big ones here. So astronauts are exposed to space radiation continuously during missions. There's no break from this radiation. And like I said before, these particles can be very, very difficult to shield, especially those GCR. And our standard radiation protection practices that we use down here on Earth, they just don't work because the three principles really are time, so limit your time. So we can do that one. We can limit the amount of time we have our astronauts up on station or on a mission. But if you're on a 3-year mission to Mars, you're on a 3-year mission to Mars. We can't really speed it up all that much, right? So we can deal with time in a way. Distance, it's all around us. It's omnipresent. So you can't really get away from it, and you can't really shield it. So they're always going to be exposed to radiation. It is completely unavoidable. And then we know that radiation has known health risks, right? But there are challenges associated with these specific kinds of health risks. One of those is that these health risks are the same outcomes that we see in the general population at very large levels. We'll take the top two for radiation, cancer and cardiovascular disease. What are the top two killers of Americans? Cardiovascular disease and cancer. So it's like finding a needle in a haystack. We're playing a statistics game that is really hard. We don't have a high signal to noise ratio, which makes things difficult. And then we also have the challenge of that, unlike a lot of the other spaceflight exposure risks, we are typically looking at long-term health outcomes, which means that our research typically takes a long time. Even if you're doing it in rodents, it still takes 2 or 3 years for those rodents to reach the end of their lifespan so we can actually look at those outcomes. And then, of course, we have a very unique radiation exposure profile. This is absolutely impossible to replicate. We can simulate it down here on Earth. We do a bang-up job of doing that, and I'll talk about that really briefly on the next slide. And then we also have limited knowledge of how these risks might be modified. The risks we know happen when you get radiation exposure. But how do they get modified by this unique space radiation exposure? We know that these ions are more damaging, but what about when they're together? Like, how's the biology react to it, right? That is what we at The Element are trying to figure out. We have, click one more. Let me see if I've got my animations right. No, go back. Thank you. We have limited observed outcomes in our astronauts. Not only are they a healthy worker population, meaning that they have just managed to stay in the workforce, indicating that they're healthy individuals, astronauts are some of the fittest people on the entire planet. So you don't expect them to have some of these outcomes. And so not only do we not have a lot of astronauts to be looking at, we don't have a lot of these specific outcomes. So our statistics, when we look at the actual astronauts, are abysmal, which is a good thing, right? We're not seeing these outcomes all that often. So we have a heavy reliance on animal studies, and this is what will get us to the brunt of what the Translational Radiation Research and Countermeasures Project is. We need to be able to translate from the animal condition to the human condition. And as anyone who has looked at any drug ever will know, how many times do things fail going from the bench to the bedside, more often than not, right? So this is a never-ending struggle of bench research. And then another thing to add on to all of these fun challenges is that it's generally a these chronic low doses and dose rates have low risks. So the risks are not that great. And so we're looking for a small signal among a large noise. So go ahead, next slide, please. So just to give you an idea of what the astronaut doses are to date, you can see here the measure on the left on the y-axis is either in milligray or millisievert. The difference between those two measures is taking into account what we call radiation quality, how much more damaging one kind of radiation is over the other. So space radiation we assume is more damaging than our terrestrial gamma radiation. So you'll see a difference between the black squares and our blue dots. The blue dots is actually their physical badge dose. And then the black squares is what we're calling our effective dose. That's sort of taking into account that radiation quality. And what you can see is these doses are relatively low. 10 milligray is or what I should say is 50 milligray per sievert is our radiation worker annual limit. So on the ground, workers in the US are allowed to get 50 millisieverts per year. That is the absolute limit. They don't want you bumping up against that limit every year. And then if just living and breathing down here on the ground as a US citizen or US resident, you get somewhere between 1 and 6 actually millisieverts just from living and breathing and being exposed to the world around you, to put that in context. So you see from our early missions of Mercury, we have slowly started to increase our dose because we're going on longer and longer missions. And you can see that Mars mission is getting up towards the 1 sievert range. Next slide, please. All right, so in terms of our ground analog, we have a fantastic facility, which is the NASA Space Radiation Laboratory, or NSRL. This is located at the Brookhaven National Laboratory up on Long Island, and it can produce single or multiple ion exposures, which we lovingly call the GCR sim. We have a sim GCR sim that is a light, sort of a GCR sim light, which is outlined here. One of them anyway, this is a 5-ion mixed field combination here. So just to give you an idea of the different ions that we use and their contribution, both in for their energy as well as their dose as the total exposures. And some links there for you if you want to learn more about the NSRL and BNL. All right, next slide, please. All right, so we've talked about some challenges, and one of our largest challenges is that we have to use animal research for the majority of our studies. We cannot ethically expose humans to the kinds of radiation doses and exposures we need to do the kind of biological research that's necessary for both characterizing and mitigating the radiation risks. So our goal here for the Translational Radiation Research and Countermeasures Project, lovingly known as TRAC, is to translate radiation research results to humans using bioinformatics and computational modeling, telling previous data generated to new sources of space-relevant radiation exposure and new animal models, including human medical radiation exposure, identify and develop preventative and in-mission therapies, and integrate countermeasures. So we have a couple of objectives. I won't read these through. You can take a look on your own. But basically, we want to take the information that we have and use bioinformatics and computational biology to see what we have and see how we might be able to apply it. Data science has just blossomed in the last 5 years, and we want to be able to leverage our previous results and that burgeoning field to see what we can get out of that data. And using this kind of data, we want to try to identify translational pathways and biomarkers that we can start to interrogate to see if we can see what's happening with these space radiation exposures, as well as develop countermeasures targeted at those specific pathways indicated in space radiation exposures. And then we want to obviously evaluate and validate those countermeasures. And these can include things for not just what you might think of as like popping a pill and now I can go hang out with the Teenage Mutant Ninja Turtles or whatever radioactive person you want to go hang out with, but things like monitoring and prevention, those things are also countermeasures. So we want to make sure that we're keeping those in mind. And then the other part of this is actually developing some digitized phantoms for larger animal models. So if we want to start using these animal models in our experiments, we know the dosimetry, especially for our different organ doses and stuff like that. All right, next slide, please. All right, so we have some challenges here, and we have a ton of data. Our first piece of the puzzle is we want to harmonize and leverage the decades of radiation research that has already been done. So the graph on the left that you're seeing is a super old, I shouldn't say super old. It's a graph, I think, from the 80s, but it looks like it's about from the 60s in the way that they used to draw those old school lines. But we've been conducting radiation research for over a century now. And we have a huge amount of different sources of radiation research over the decades. But compare that picture on the left to the kind of data we're getting now. This is genotyping data coming from an outbred strain, and they're looking at specific hits. I mean, it's incredible, this kind of data. So how do we get this data across these decades of research? Oh God, my computer's gone to sleep, sorry. How do we get these different data sources from across the decades to harmonize them? How do we do that so we can leverage all of the decades of data? So that's one piece of the puzzle. Next slide, please. The next piece is our animal models. And so what you're seeing here is this is a great paper that put together all the different neurobehavioral and cognitive tests and studies that have been done on this great chart, because it really shows you how much data is out there and what you're seeing and what's showing no impairment and what's showing impairment and the different kinds of stuff. And there's stuff all over the place, right? So again, how do we bring all this animal model data together? So that's another piece. Next slide, please. And so ionizing radiation is something that we use in humans actually all the time. We are unique in radiation that we actually do have a lot of data in humans. And we continue to use ionizing radiation for very beneficial purposes. So we have human medical data sets. So how do we leverage this? But this kind of data is totally different than the data we're normally used to seeing in our animal models. So this is another piece of our puzzle. Next slide, please. And then we have individual susceptibility. This is the same paper that I showed you with the genomic data, looking at different families of mice and seeing where the tumors are popping up from different irradiations. And you see that the tumors, and this is an outbred stock of mice, you see that the tumors are really localized to family groupings, indicating that genetic susceptibility is a big driver here and not so much the radiation type, which is really interesting. So that's another piece of the puzzle. So next slide, please. So the point of TRACK is to bring all of those different pieces together. And like I said, I just started in April. So next slide, please. So really where we are right now is we are just at the beginning of this journey and figuring out how we're going to harmonize and leverage all of these different disparate radiation research sources to bring this data together and create a really robust platform with the ultimate goal of informing both risk characterization and mitigation for radiation-associated health outcomes. So that is all I've got for you today. So next slide, I'm happy to take any questions once we go to Q&A, but I do want to make a shameless plug for a couple of our different solicitations that we have live right now. We have one for countermeasures for RFI, and we have a couple more out there as well. So be sure to check out our stuff out on NSPIRES. So thank you so much.
Thank you very much, Robin. And I think Jennifer Buckley, are you going to moderate the questions and answers now?
Yes, we can go ahead. We have a few minutes left in the session, and we'll try and go through a couple quick questions. Thanks for sending those in. The first question that we have is going to be for Jen, and that is, do astronauts take any probiotics, or is that something considered at this being considered at this time?
So interestingly enough, we've had both people express an interest. I mean, astronauts work with their flight surgeon on any kind of medications or supplements that they're going to take. That's important metadata for anyone doing experiments on astronauts, of course, to know and to figure out if they can factor it into whatever it is they're trying to observe. JAXA actually had a very deliberate probiotic study where they were looking at certain commercial brands. So yeah, it does happen. I mean, I think, you know, when you look at the terrestrial literature and you try to understand the role of gut health and increasing and making sure you have a healthy diversity in your microbiome, it's important. It would be really great if we could quantify it and then also consider how the probiotic might be changing over time due to spaceflight stressors. So any kind of supplement or pharmaceutical is not impervious to change over time due to things like what we're accustomed to on Earth, which would be humidity or exposure to air and light. But then, of course, we have some unique stressors. So in terms of safety, we would want to do a more methodical study of it, and we certainly have those plans, but it's introducing another variable at this point. So I think it's out there. I think we have ways to kind of tease it out, but it should be a very deliberate effort. And we do have some analog studies where probiotic use is a very deliberate effort as a complement to the food system.
Okay. Thank you, Jen. Next question for Bill. How do you study gravity gradients and the transition between different gravities?
We've done a lot of things over the years, what we're mostly doing these days is looking at crew members, usually when they return to Earth, and looking at somebody who's been in flight for, nowadays it's usually 6 months. Back in the shuttle era, it was a couple of weeks. Mir station, it was 3 months. And look at their performance immediately after flight compared to what it was immediately before flight, so that we can understand the impacts of the transition coming back to Earth once they've adapted to the microgravity environment. During the space lab era and early in the space station, we did a lot of rodent experiments and some other toadfish experiments to look at adaptation of the vestibular system to going uphill to looking at what was going on with them going into space and their adaptation. And there have been a number of rodent experiments doing the same thing. Today we're not looking at that heavily. We're looking mostly at the performance effects of that. We've been doing what we call the field test at the Soyuz landing sites and looking at crew members right after they land. I think we're going to start doing more of that when we have the commercial providers bringing people back and landing them on Earth in the U.S. In the future.
Okay, next question, we'll go to Steve. Is SANS reversible, or do you see permanent changes?
That's a good question. Most of the crew members who have been studied, it seems to be reversible, although there have been a few crew members where they have lasting effects that have gone out at least 6 months. Most of our studies end at 6 months. A few have gone to a year. And so more of our studies are taking a longer term approach, looking at recovery. Now, when I say that too, please remember that most of the studies that have been done have looked mostly at the eye and have not looked at the brain or any other issues there. So we know that for the most part, vision recovers with a few folks who don't, but we don't know about any other issues besides the eye. And those studies are being solicited for, and some are in the process of going on right now. So we hope to have a better answer for that question in the near future.
And for our last question, we'll go to Robin. Is there a particular activity or time in transition that is considered the most risky for radiation?
That's a great question. So the typical space radiation environment is that chronic low-level GCR that is just constant. So the risk is pretty flat across the board. And then also, I want to clarify before, the risks in mission are relatively low because the largest change in our local radiation environment is due to those solar particle events. Those initiate from our sun. They're really unpredictable. And so in order to counteract that, what we have operationally done is developed what's called a storm shelter. And we should call it a radiation storm shelter because otherwise it's like, but there's no hurricanes. So what that is it's you use mass that is already on board and you reconfigure the habitable volume to build yourself a little bunker. And you hang out in there until the storm has passed, the particle storm that is. And that there are requirements on the books that we have to be able to build those storm shelters so the level of radiation doesn't get above a certain threshold. And that is to prevent any in-mission acute radiation symptoms. And so that would likely be the highest risk time for an astronaut is during one of those solar particle events. But those risks are optimized, or not, the risks aren't optimized, the mitigation strategies are optimized to keep those risks as low as reasonably achievable. So that would probably be the highest riskiest time.
Okay, thank you, Robin. And thanks to everyone for submitting your questions. We don't have any time for any additional questions today, but I do want to thank all of our speakers for their excellent, very informative presentations. So thank you to Bill, Jen, Steve, and Robin. And that concludes our session.